Hello guys, welcome back. This is Dr. Hadi here, and today's topic is again from the biochemistry, from the chapter enzyme. This chapter is getting more and more interest as we are proceeding, we are progressing in this chapter. Few lectures was already recorded about enzyme, the classification, the history. Today's topic is class and subclass of enzyme. Actually, this topic is also classification of enzyme, but it is in detail now, right? We have already discussed the classes of enzyme, but not in depth. So in this video, we will discuss the 6 classes of enzyme, and within the 6 classes, the subclasses of enzyme, each one by one, but in simple words, right? The classes and subclasses of enzymes are 6 classes. The first one is 6 classes. Oxidoreductases, this is one class. Then Transferases, second, and then third Hydrolases, fourth one Lyases, fifth one Isomerases, sixth one Ligases. All of the enzymes throughout the world, all of the enzymes, maybe thousands and thousands, all of these enzymes belong to any of these groups. You will not find a single enzyme that is not belonging to any of these single groups. Fine. So whatever enzyme you have or you have seen somewhere, these enzymes must be from one of these groups. Let's discuss the first group, that is oxidoreductases. The first group need little bit more discussion, a little bit more time. Second group also need little bit more time, but the third, fourth, fifth and sixth group, they are very simple and they don't need that much time. So we will be able to finish them as soon as possible. Oxidoreductases. Most of the students, they know very well what is oxidation and reduction. Still, I'm going to tell you, I'm going to discuss the oxidation and reduction. Here, what do you mean by oxidation? If you have any substance, if you have any compound, if you have any molecule, so oxidation will be, the first definition of oxidation will be oxidation. What is oxidation? If you add oxygen into it, that will be called oxidation, the first one. And if you remove hydrogen from it, look, the arrow for outside the box tell that you are removing hydrogen. This is removal of hydrogen. So if you remove hydrogen from a substance, this is also called oxidation. And if you remove electron from a substance, from a molecule, electron is denoted by E, this is also called oxidation. So we can define oxidation in 3 ways: addition of oxygen, or removal of hydrogen, or removal of electron. This is called oxidation. Now the second thing is what? Reduction. Now what is reduction? Reduction is the opposite of the oxidation. Means you have any substance or molecule, when you remove oxygen from it, this is called reduction. It is the opposite of this. Here you are adding oxygen, but here we are removing oxygen. If you add hydrogen into it, this is also called reduction. If you add electron into it, this is also called reduction. So oxidation and reduction are the opposite of one another. These oxidation and reduction are most important reactions that are taking place in our body, in the living cell, in the living body. So there are enzymes, not enzyme. There are several enzymes which are which are involved in these type of reactions, in oxidation and reduction. So all of these enzymes which are involved in oxidation and reduction, they are called oxidoreductases family. We made one big class or family. This one big class or family is named as oxidoreductases, just to discuss these enzymes. Okay, so we have oxidoreductases. Now the definition is clear now. You can very easily define that. You can easily write the definition of oxidoreductases, but if you want to go into the depth, a little bit into the depth, you want to explore a little bit more about oxidoreductases, then you need to go into the depth. Means you need to study the subclass. This is just one class, and there are subclasses. Just like you say in the world, there are many people are there, right? We have human population. You say classify them, so you will make 2 groups or 3 groups. Means Asian people, African, South African, American. This is one class. Then within Asian, you say we can classify these people further. So within Asian, you will come Hindi, Indian, Pakistani, Chinese. So within one group, you make another small group. Similarly, in the class of enzyme, we make some small classes that is called subclass. So all of them belong to oxidoreductases. So what are the subclasses? These are number 1, oxidases, aerobic dehydrogenases, anaerobic dehydrogenases, hydroperoxidases, oxygenases, and reductases. So these are 6 subclasses within single oxidoreductase class. Let me discuss one by one. Oxidases. These are those enzymes which are involved in the addition of oxygen. They add oxygen. What do they do? They add oxygen, but in what? Into hydrogen removed from the substrate. Look, this is a substrate. You just don't focus much on the structure. This is just a substrate, and it is called as ascorbic or ascorbate. Ascorbate. I have not written the complete structure of ascorbate. And today's topic is not about the structure of ascorbate. The topic is about the enzyme. So the main idea, our main focus should be on the enzyme, not on the complete structure. So you may find some groups missing in that structure, which we don't care about. So here this is ascorbate related structure. In this structure you have OH and OH, right? What actually happens? We remove these 2 hydrogen. When these 2 hydrogen are removed, fine, these 2 hydrogen are removed. So with these 2 hydrogen, we will add what? We will add oxygen. Now this red color oxygen, who add these enzymes? Oxidases. This addition of oxygen into that hydrogen which came from the substrate is called oxidases. So now, of course, the hydrogen oxygen will combine to form H2O. Fine. And what will happen to the substrate? Substrate will become, here you will find one double bond, and here you will find another double bond. Fine. You also don't worry about the structure double bond, but at least we got the idea that from the substrate hydrogen atoms are removed, and these hydrogen atoms were given oxygen by the oxidases. So this is the style. You must remove your confusion by memorizing the mechanism of the oxidases, because when you go further you will get some confusion if you are not clear here. You should focus that what happens, did some kind of oxidation reduction take place here? So yes, you say yes, oxidation took place here because the hydrogen removed from the substrate, this is oxidation, and the enzyme added oxygen into that hydrogen, this is also oxidation. So what we got, this is called dehydroascorbate. Dehydro means hydrogen has been removed, so this is called dehydroascorbate. You will write this example under the heading of oxidases if you are in the examination hall. Of course you are not watching my video in the examination hall, you may be in your class or room. Aerobic dehydrogenases, this is second subclass of the oxidoreductases. These remove hydrogen from the substrate. Now look, see, these remove hydrogen from the substrate. These were not removing hydrogen from the substrate, they were adding oxygen into the hydrogen. these will remove hydrogen from the substrate. This is what students face, this kind of confusion every time. So definition, they remove hydrogen. They remove hydrogen, they add oxygen. Oxidases, they add oxygen. Dehydrogenases, they remove hydrogen. So they remove hydrogen. Let's suppose this is glucose. Again, I have not mentioned the complete structure of glucose. I don't need to write the complete structure of glucose. And most of my students, they are very good students. They already know the structure of glucose. And so I don't need to write the structure. The main idea should be on the main groups. The focus should be on the main groups. So this is the structure of the glucose. You have oxygen, carbon number 1, hydrogen, and OH. Now with this, you can see here, here you will find hydrogen and here you will find hydrogen. These two hydrogen will be removed, right? When these hydrogen are removed, here at carbon you will find only oxygen. This was OH in the beginning, and now it become just oxygen because the hydrogen was removed. And who removed the hydrogen? Aerobic dehydrogenases. One thing more, once the hydrogen atoms are removed from the substrate, मैं थोड़ा सा तेज बोलता हूँ, so maybe उर्दू स्पीकिंग वालों के लिए थोड़ा ये मसला हो, but be in mind, bear it, right? कोशिश करता हूँ कि थोड़ा सा उर्दू भी बोलूँ साथ साथ, right? I will try to speak some Urdu as well as it is in English, but a little bit Urdu will be there. Okay, so you should focus on when the hydrogen atoms are removed, then what happened? Is there any acceptor? Means is there any other, once these hydrogen are removed, where these will go, these hydrogen atoms, where? There will be some sort of acceptor. There will be some sort of what? Acceptor. There will be something to get these hydrogen. Means you have hydrogen, when these are removed, they should go there. So there should be an acceptor, and that acceptor, in this case, in the case of aerobic, look, aerobic is for oxygen, aerobic is for air. So in this case, the acceptor is oxygen. Means the hydrogen which are removed from the substrate, they will go to the oxygen. So oxygen will get the hydrogen, and once again oxidation took place. Fine. The acceptor is most important here, because the next category, the next subclass, which is called anaerobic dehydrogenase, it is also dehydrogenase, and this one is also dehydrogenase. What will be the difference between these two? This dehydrogenase will remove hydrogen. This dehydrogenase enzyme will also remove the hydrogen from the substrate. This is substrate, this is substrate. But in this case, the hydrogen which was removed, it will be given to another acceptor. But this time, there will be no oxygen. This time, the acceptor will be, NAD, nicotinamide adenine dinucleotide. It's a kind of coenzyme. It's a kind of cofactor, came from vitamin. Yes. And you know very well about coenzyme, cofactor, right? So this, in this case, the hydrogen will be given to NAD. Anaerobic, without oxygen. So anaerobic dehydrogenase, they will oxidize, they will perform oxidation, but the hydrogen which were removed, they will be given to whom? To NAD. Look at the example we have. This is a structure of lactic acid, you can say lactic acid. Fortunately, this time the structure is complete for the lactic acid. Yes, it's a complete structure of lactic acid. If you notice here, this carbon has hydrogen at the top and OH below it. So if you see here, this lactate or lactic acid is converting into what? Into pyruvate. Right? This is what? This is pyruvate, you know, the product of glycolysis, pyruvate. Pyruvate do not have that hydrogen in this OH. However, the pyruvate have the oxygen, but these two hydrogen atoms are gone. These two hydrogen atoms mean this structure is converting into pyruvate. Where these two hydrogen will go? These two hydrogen will be received by the NAD. So NAD becomes what? NAD becomes NADH. NADH. This is oxidized form. This is the reduced form. You can write another H as well. You can write another H as well, but you will write a plus sign. But don't worry. The main focus thing is that hydrogen. This is oxidized. This is reduced. So this is another enzyme, anaerobic dehydrogenase. The fourth one is hydroperoxidases. Hydroperoxidases. At the end of video lecture, if I remind that, I will go towards my camera and I will zoom it for you, the whole lecture, right? If for some students the fonts are little bit small, अगर बाज़ students के लिए छोटे नज़र आ रहे हैं, तो मैं lecture के आखिर में इसको zoom करूँगा. Fine? So that you can note all these points on the whiteboard. Now the hydroperoxidases, these are, what sort of enzyme these are? Hydroperoxidases, these enzymes are involved in the formation of breakdown of hydrogen peroxide. There is one compound, hydrogen peroxide, H2O2, hydrogen peroxide. All of these hydroperoxidases, they are involved, they are linked with this compound, hydrogen peroxide. Within this category, we have two types of enzyme. One is called catalase, otherwise called peroxidase. So catalase and peroxidase, these belong to the hydroperoxidases. What catalase do? A catalase break down hydrogen peroxide into water and oxygen. If you see here, there is oxygen and there is hydrogen, H2O. And peroxidase enzyme, they do what? H2O2, again we have hydrogen peroxide, but this time H2O2 is given 2 electrons, 2 protons, and by the addition of these 2 electrons and 2 protons, the H2O2 becomes just H2O. So this is the difference between the catalase and the peroxidase. If someone say, if a question comes in exam that differentiate between the catalase and peroxidase, if this is just a short question, what would you write? You will draw one single line in the middle of the page, and on the right side you will write catalase, and on the left side you will say peroxidase, or vice versa. Below these two you will write catalase is involved in the breakdown of hydrogen peroxide and the product is water and oxygen, while in case of peroxidase, the peroxidase break down hydrogen peroxide, but this time it breaks down the hydrogen peroxide into water only. So this is the main difference between the catalase and the peroxidase. The other similarity between these two is that both catalase and peroxidase are hydrogen peroxidases. Both act on hydrogen peroxide. This is the similarity. So you got one short answer as well. Then we have oxygenases. Now look at that. There is oxidases. Oxygen. There must be some difference between the oxidase and oxygenase, and I also told you in the beginning of the video lecture that you should give focus on what I am telling you here, because you will feel some difficulty then. But if you focused, if you were attentive here, you will feel no problem here. Yes, that's my promise. So oxygenase, what oxygenase do? They incorporate molecular oxygen to the substrate. They incorporate molecular oxygen. What are they doing? They add oxygen into the hydrogen indirectly. They add oxygen. They were adding oxygen, but not into the substrate. To the hydrogen that were removed from the substrate. To the hydrogen that were removed from the substrate, and then to this hydrogen they were adding oxygen. But this time they are adding oxygen directly into the substrate. So here what they say, incorporate molecular oxygen to the substrate. The example is we have an amino acid, phenylalanine. Again, the structure is not complete. Only the part that of interest has been shown. Yes, this is our interest, phenylalanine. You know, this benzene ring, and there is C double bond OH, carbon, amino group, everything is there. So in phenylalanine, you can see there is no OH, but once the oxygen is added, yes, oxygen is added, it become what? It become tyrosine, means you will find oxygen in the form of OH. You will find oxygen in the form of OH. This is the oxygen that was added to that structure, phenylalanine, and as a result of this addition, phenylalanine become what? Become tyrosine. Here, of course, NADH will also be used. Yeah, this reduced NADH will be used in that reaction. Okay, sometime if you see an arrowhead like this, it means that only this is possible, the conversion of this into that. And if you see another arrow like this, it means the reactions are possible, reversible. Okay, now the sixth one is the last subclass, is the reductases. Reductases, what they do? Okay, all of these, they were involved in oxidation. Some sort of oxidation was there. Oxidation was there. Different style of oxidation. But this is the subclass of enzyme that is not involved in oxidation. It is involved in the reduction. And you know very well what is reduction. Reduction is the addition of hydrogen, the addition of electron, or the removal of oxygen. So here you can see there is a structure called glutathione. I hope you can see this structure, glutathione. This glutathione is actually what it is. Glutathione is actually a combination of 3 amino acids. Let me write just simple 3 amino acids. This is 1 amino acid. Fine. This is another amino acid, and this is 3rd amino acid. Okay, I'm again going to write these 3 amino acids linked together. So when 3 amino acids are linked together, they form what? They form glutathione. This time, once again, there is no need to find the structure of this amino acid and that. You just notice here, these are 3 amino acids. They are linked and they are called glutathione. Once you become mature, of course, when you become mature, you will find the structure of these 3 amino acids. Okay, in case of this one, if you find a OH here, all right, SH or OH here, so this is called reduced glutathione. Focus on that hydrogen. Maybe you see an OH or H. This hydrogen is there. This is reduced, and here there is no hydrogen. So this is what? Oxidized form. So oxidized form of glutathione and reduced form. Now there is any enzyme which is involved in the reduction of the oxidized glutathione into the reduced glutathione. That enzyme is called what? Reductases. So here is the example, glutathione, right? Glutathione oxidized, glutathione reduced. Here I have mentioned H. Means in the glutathione you have what? You have hydrogen. Okay, so this was a complete lecture on the oxidoreductases. Now come to the next one, transferases. Transferases, these are those enzymes which are involved. These are not involved in oxidation reduction. Fine. These are involved. They have some new style. They are involved in a lot of reactions, chemical reactions, but their style is a little bit different. They transfer one group from one molecule to another molecule. So if you have, this is like, let's suppose if you have, this is one molecule and this is another molecule B. So what they will do, let's suppose they will transfer a small group, let's suppose X, from this here to this one. So it's called like a kind of transfer, maybe in exchange, sometime in exchange, transfer from this one here and from this one here. So this kind of transferase enzymes they are transfer the groups from one molecule into another. Now based on this transfer, the different groups can be transferred, different groups, like phosphotransferases. If it is phosphotransferases, so of course these enzymes, they are transferring phosphate. If it is transaminases, so it means they are involved in the transfer of amino group NH3. If it becomes NH2, it becomes amino group. Fine, amino group. Otherwise, it is ammonia. Transmethylase. These will be the enzyme which are involved in the transfer of the methyl group, right? And transpeptidase, they will transfer an amino acid. They will transfer an amino acid from one group to another, or just a peptide, means many amino acid. Yes, many amino acid linked together. These enzymes will transfer many peptide from one place to another, or just one amino acid from one place to another. So these are called transpeptidases. And then transacylases. Transacylase, these enzyme will transfer acyl group. You know what is acyl group? Acyl group is actually, this is CH3 C double bond O. This is always called the acetyl group, or we call acyl group. So these are involved in the transfer of the acyl group. For the convenience, in order to make my video short, not that much lengthy, I would just focus on phosphotransferases only. Phosphotransferases. Phosphotransferase example is available here. Look at that, that is a glucose. That glucose has carbon number 1, 2, 3, 4, 5, and this here we have carbon number 6, right here carbon number 6. By the help of ATP, you know, in ATP we have how many phosphate? In ATP we have 1, 2, 3 phosphate. In ATP we have 3 phosphate. So with the help of ATP and that enzyme phosphotransferase, the phosphotransferase enzyme will take 1 phosphate and add into that glucose, add into that structure, transfer it. Look, ATP is 1 molecule, so a small part of this molecule, that is 1 phosphate, it is transferred from this ATP to that glucose. As a result, you will find here the phosphate PO3 here, it is attached to the glucose. So now it is just glucose. It is just what? It is just glucose. Right? You can call it D-glucose, but it is glucose 6-phosphate. Glucose 6-phosphate. Glucose 6-phosphate. Now, who did this? This is called phosphotransferases, and such enzymes are also called kinases. They are also called kinases. So in biochemistry, may you find some kinase, the word kinases. Kinase enzymes are involved in the transfer of the phosphate group. And so this was a wonderful example, and this example was already discussed in my previous video lecture where I gave a summary of the classification of the enzyme. And then another example is also available, that is transmethylase. Transmethylase, one of good example is norepinephrine and epinephrine. You know the two words epinephrine and norepinephrine. I have also recorded a video lecture on the neurotransmitter. You can watch that video lecture. So epinephrine and norepinephrine, these two are neurotransmitters. These are two other hormones as well. Epinephrine have in its structure, like if I write here R, there is NH and there is CH3 group. So when CH3 is there and it is attached to the amino group, it will be called what? Epinephrine. If we just remove this NH, means R is there, NH is there, but there is no CH3. When there is no CH3, it will be called norepinephrine. So look, when it is norepinephrine, it has just NH2 group, and when we transfer a methyl group here, it will become epinephrine. Maybe the reversal is there, maybe the methyl group is the part of the norepinephrine or the part of the epinephrine, but the main idea is that you should focus that what is going on. If methyl is transferred, it's called methyl transferases. Okay, so the good example was this one. You just don't focus on that one. It was just an example that came into my mind this time, right? This is on the board. Okay, then we have the hydrolases, the third category. Okay, one more example is there, acetylcholine and choline. Acetyl-CoA, that is acetyl-CoA, CH3 C double bond O S-CoA. Fine, this is what acetyl-CoA, and choline is another group, and a nitrogenous base. So when you add this acetyl-CoA into choline, choline is a nitrogenous base, you will get what? You will get acetylcholine. Acetylcholine is a complete nitrogenous base. Now you can find this in many lipids. Now hydrolases. Hydrolases are those enzymes which are involved in the breakdown of compound by the help of water. So I will erase this little bit. This place, I need some space here to write ideas, to write something for you. That is, I have to do this. Okay. Okay, now hydrolases, the mechanism, what they do, how they break down a compound. Like, let's suppose this is a compound, and is linked, two part. Now by the help of water, H2O, H and OH, by the help of water, one of the hydrogen will attach here, the OH will attach here to that part, to that segment. Suppose this is one molecule, it has two segments. So as a result of this OH attachment here, this bond will be broken. And what will happen? You will see one part, one segment, and another segment. One segment would be having the hydrogen, another segment would be having the OH. Now these two segments were broken. And now who did this? Water did this. Water did this. And the breaking process by the help of water is called hydrolysis, and there must be some enzyme. If any enzyme is involved in such kind of reaction, if any enzyme is involved in this kind of breakdown, these enzymes will be called hydrolases, and there are several hydrolases which are performing hydrolysis. Proteases, these are those enzymes which act on what? These enzymes act on the protein, right? You know, in protein we have one amino acid linked to another amino acid. There is a bond between the two amino acids. That bond is called what? That bond is called peptide bond. Which bond? That bond is called peptide bond. And the peptide bond is always CN bond. Always which bond? CN bond. This we always write CN bond. So between the two amino acids there is what? CN bond. I'm not going to write the complete structure of the two amino acids because my video lecture is already available on the structure of two amino acids, how the two amino acids link together, everything is available there. So the main thing is that we should focus on the groups of interest only. So protease, they hydrolyze this bond. So that carbon become become the part of this amino acid only, and that nitrogen become the part of this, and as a result the two amino acids you separate. In this category, in this time, these two amino acids are linked, but after hydrolysis, after the addition of H and OH, the two amino acids will be separated, and these enzymes are called proteases. So proteases, they will break peptide bond, they will hydrolyze peptide bond. In carbohydrates, there is one glucose. Let's suppose there is one glucose. I will just write GL, glucose, and there is another glucose. I will write GL, two glucose. There is no need to write the structure of the glucose. There is bond that is COC bond. In case of glucose, there is COC bond is there. Like in amino acid, protein, CN bond is there for peptide bond. COC bond is for glycosidic bond. So this bond can also be broken just like the peptide bond by the addition of water. How the water will be added, just already explained. So once the addition of water, the bond is broken, this glucose and that glucose will no more be the part of each other. They will be acted as a separate units, one glucose and a glucose. So this is called carbohydrates. It include what? Those enzymes which act on starch, which act on sucrases, lactases. All of these enzymes that you know, lactase, sucrase, these are actually carbohydrates, and these are actually hydrolases. Then lipase are kind of lipids, and in lipids, you know, there is what? Fatty acids. Fatty acid linked with what? Linked with glycerol. Linked with what? Glycerol. My video is available on that topic, how glycerol is linked with fatty acid. You can check lipids by Dr. Hadi, ester formation by Dr. Hadi. The video are available on all of these topics. You can just go there. So when fatty acid is combined with glycerol, there is a bond will be formed. That bond is called ester. That bond is called ester. So you can also break down this bond by the help of water. So if we break this bond, this is called lipases. This can also be called esterases. This can also be called what? Esterase and lipases. right? So lipase and esterase, the definition is so clear now. Lipase and esterase, these are the enzyme which hydrolyze, which bond? Ester bond in the lipids. Fine. So we saw that these all categories are involved in hydrolases. Now we have the fourth category, that is Lyases. As I told you in the beginning of the video lecture, the remaining subclass will take less time as compared to the first two classes. The Lyases enzyme, they will catalyze the addition of these groups. They will add groups. They will not transfer, they will not hydrolyze, they will add groups. Which groups? Most probably the amino group NH3, water, or the CO2 groups. They add these groups, one of these, one or that one or this one, add into a compound that has double bond. For example, there is a compound, and you should not be worried about this. This is a compound. Yes, it is a compound. This is one carbon. This is another carbon. You can write it here in simple words. Some students, they know very well. But this one is actually carbon. Here is another carbon. And there is double bond. Then there is carbon. Right? Yes. This one. Fine. So carbon, double bond, carbon. Yes, this is a compound, but for the simplicity we just write it. Only the double bond has been shown. And the name of this compound is called fumaric acid. You know, the part of a Krebs cycle, right? Krebs cycle. Fumaric acid has a double bond in between the two carbon. And now we can add what? We can add water here. This is H2O. This time the H2O is not going to hydrolyze the compound. This time the H2O is not going to break this bond. This time the H2O will broken down into H on OH. The H of the water and the OH of the water. H will come here, OH will come here. So look here, you can't see any double bond here because that double bond is consumed. ये डबल बॉन्ड इस्तेमाल हो जाएगा जब आपका ये वाटर इधर ऐड होगा तो डबल बॉन्ड इस्तेमाल होगा. द डबल बॉन्ड हियर विल बी यूज्ड once that water is added, because double bond is a very good place for addition, something to add. If you want to add a group in a double bond in a compound, you should see a double bond there. So double bond is a very good place for addition. So there is no double bond here. However, in place of this you will find H and OH there. So this is now called what? Malic acid. This fumaric acid double bond compound is called fumaric. The same compound with single covalent bond and H and OH, this is called what? Malic acid. So the enzyme which perform this kind of activity, they are called lyases. So lyases will not only add water molecule, lyases can add ammonia molecule, amino group. Lyases can add carbon dioxide as well. I have just mentioned this example of fumaric acid just to tell you that these enzymes add a group in that compound, and this can be reversible as well. Means if you reverse it, from the malic acid you remove hydrogen and OH, and then again a double bond will be created. This is the reversal. And you know, the interesting thing is that lyases are involved in both. Lyases can add a group to a double bond making it single bond, or remove that group making it double bond. So lyases are involved in both type of reactions. You should notice it, you should note it. And whatever I am telling you, you can write, you can note this on your register, and you will be having a wonderful marks in exam if you just write these whole lecture on at least 6 pages or 5 pages. You would be having so much to write on that paper. The fifth one is isomerases. Isomerases, these are those enzymes which are involved in the formation of isomer. You know, isomer, we know very well. What is isomer? Let's suppose you have one compound, carbon, here another carbon, right? Here is OH, and here is H, and there are CH3. Fine. Okay, and here you can also have CH3. Isomer, what is isomer? If I see this OH is here at that carbon, and if that OH comes here on that carbon, fine, let's suppose, that become CH2 and it becomes CH2. Fine. Now this is similar compound. Both have same formula, but only the difference of OH group. So this is called isomer. A very good video lecture of mine is already recorded on isomerization process, isomers. You can watch this video. But you know what I'm going to tell you, there are some reactions where we have one compound A that is converted into B. Both of these are isomer of one another. Both have same formula. So there are enzymes which are involved in the process of isomerization. Let me give you one example of that. Look, the example of isomerase one is phosphohexose isomerase. There are so many enzymes, but one example is phosphohexose isomerase. What it do here? This is a glucose. This is a hexose. Fine, it's a glucose. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, 5, and 6. At the 6th position, there is P, means there is phosphate. So what name can you give it? This is called glucose 6-phosphate. Yes, this is glucose 6-phosphate. It is in front of you. An enzyme will come, and this enzyme will do what in that structure? You see here. That enzyme will just bring some changes here, like now see here, this is carbon number 1, this was carbon number 1 here in the first case. It was 6 corner ring, now it become 5 corner ring. Carbon number 1 is still there, carbon number 2 is still there, carbon number 3, 4, 5 and 6, carbon number 1 to 6 all are there. Even still we have phosphate group. At 6 we have the phosphate group is also available, but the difference is that this is glucose 6-phosphate. This is what? This is fructose 6-phosphate. Glucose 6-phosphate and fructose 6-phosphate both have same molecular formula. Both have 6 carbon, 12 hydrogen and 6 oxygen. You can see my video lecture on that as well. The difference between glucose and fructose. I have recorded so many videos on monosaccharides. I have recorded a lot of lectures on carbohydrate. You can find this. So this is the difference between glucose 6-phosphate and fructose 6-phosphate. Fructose is 5 corner ring. It is a kind of a ketonic sugar, and glucose is an aldehydic sugar. So what happened? The glucose became fructose. Fructose and glucose are isomer of one another. And which enzyme did this? The enzyme phosphohexoisomerase did this. So there are a lot of other enzymes also available performing this kind of action. The last one is ligases. Ligases, these are those enzymes which are not doing any work of the 5 groups. They have their own style. They are not hydrolyzing, they are not adding, they are not oxidizing, they're not reducing. They just link 2 molecules together. They link. Now the word they link 2 molecules together you will not find in the previous 5 groups. They link 2 molecules together. Let's suppose one example we have, that is the DNA ligases. DNA ligases. This is one example, DNA ligases. In DNA ligases, we have one nucleotide. This is one nucleotide. Here we have another nucleotide. Okay, many nucleotides are linked together in the DNA. Nucleotide. Once again, my video lecture is also available on the DNA and RNA. Especially a very good lecture on the structure of the DNA is also available in Urdu as well as in English. Fine. So I hope you know very well the structure of the DNA. In DNA we have many nucleotides. During the synthesis of the DNA, when DNA molecule is synthesizing, we have one nucleotide here, and at this nucleotide we have a phosphate. Then we have another nucleotide here. Then we have another nucleotide here. Okay, it has OH only. Fine. So we want these 2 nucleotides to link with one another. We are going to link them. Fine. So an enzyme will come that is called DNA ligase. The DNA ligase, what it will do? The DNA ligase will make a bond between that oxygen and this phosphate. This OH is at carbon number 3. Let me write it a little bit large size. You can just pause the video and take a screenshot. I'm going to write here something for you. This is one nucleotide, this is one nucleotide, and this is another nucleotide. This nucleotide has phosphate group, and this nucleotide has OH group. It is carbon number 1, 2, and 3. Most probably it is the OH is at carbon number 3, and this is the phosphate group connected with another nucleotide at carbon number 6. So the enzyme, they will make a bond between that phosphate and this oxygen. The hydrogen will be removed then. Hydrogen will be removed. This bond is called phosphodiester bond. Diester bond. Of course, there's one bond is already here. The two bond make diester. It is actually a kind of ester bond. So the two molecules were linked together by the help of what? By the ligase. So the DNA ligase, you can find this concept in my video, DNA replication. A very large video, a very large topic, a very good topic is there on the DNA replication. Within the DNA replication, you will find DNA ligase. I hope this was the complete lecture about the class and subclass of enzyme. It was a little bit lengthy. I hope those students who were getting interest in that, they were getting something, they would watch this lecture from the beginning till the end. At the end, I would like to share one thing with you, that always think positively about yourself and about others. If you hear something bad from someone else that is blaming on other, you try to shut this thing within your brain. You try not to spread this blame. Maybe anything that you heard, maybe it was not right, maybe it was false information, but you spread it to others. So try to be more positive, as much positive as possible. Thank you for watching the video. I will zoom it. Yes, I just memorized. And thank you so much for watching the video. Allah Hafiz.